Good afternoon, everyone. My name is Teti. I'm the chair of this webinar that will be presented by Dr. Silvia Marino. As you can imagine, she's Italian, and she's Associate Staff Scientist in the Department of Physiology and Cell Biology at the University of Arkansas for Medical Sciences in Little Rock, in Arkansas, US. Silvia graduated in Turin in pharmacy and got the PhD in experimental medicine and therapy in Turin. Then she moved to the University of Edinburgh and then to Sheffield under the supervision of Dr. Eamon Idris and Professor Stuart Ralston. And here she investigated cancer and bone. Then she moved to the Indiana University in Indianapolis in the US under the supervision of Professor David Rudman and Teresita Bellido. And she investigated, worked on hematology and oncology. And now she's at the University of Arkansas, where she's working on bone pathophysiology. Silvia has a lot of studies performed on different topics. For instance, she investigated the role of IKKB and FKB pathway in the bone metabolism, bone metastasis, the role of endocannabinoids and their receptors in the regulation of cancer-induced osteolysis in bone metastasis and primary bone tumors. She investigated the role of SQSTM1P62 as a druggable target for multiple myeloma, and now she's working on bone diseases induced by diabetes. She received several honors, especially when she presented in international meetings, including the European Cancer Tissue Society meeting and the American Society for Bone and Mineral Research meeting. She's supported by several grants from the American Society of Hematology, from the National Institute of Health, the Sturgis Foundation Award, and the Multiple Myeloma Research Foundation. So we are very happy to have you here. Silvia, the title of the webinar is Reversal of the Diabetic Bone Signature with Anabolic Therapies in Mice. Silvia, the floor is yours.
Thank you so much, Professor Teti, and good afternoon, everyone. And thank you for the opportunity to be here with you today. So, and as Professor Teti said, today I would like to present you the data of a project that I've been involved since I joined the laboratory of Dr. Bellido here at UAMS in 2021. So Yes. So I promise Professor Teti some slides of my sort of like life and scientific life, and I would like to show you a picture of how in a picture way, how I end up here. So as I started my PhD in the beautiful Turin, in the laboratory of Professor Francesco Di Carlo. And here I studied the effect of PTH or teriparatide on fracture healing. During this time, I spent a few weeks in France, in Lyon, in the laboratory of Professor Georges Boivin. And here I did some mechanical testing the hardness of the callus in the animal, the newly callus forming the mice treated with PTH. Then I moved to the beautiful Edinburgh in Scotland in the laboratory of Prof. Stuart Ralston, where under the mentorship of Dr. Eamon Idris, I studied the interaction between solid tumor, mainly breast cancer, and osteoclast, the bone resorbing cells, and with the mean of identifying new druggable target to halt bone metastasis. And then I follow Dr. Idris at the University of Sheffield to continue my work on bone metastasis. And while I was here, I visited the laboratory of Professoressa Teti, as we were having a very successful collaboration with Professoressa Rucci. Now in 2017, I moved to Indianapolis, to the States, in the laboratory of Dr. Rudman. And here I study how multiple myeloma induced bone disease. And I focus now this time, I abandoned the osteoclast, and I focus this time on osteoblast, the bone forming cells. And I studied the mechanism by which multiple myeloma induced osteoblast suppression. And then in 2021, 1.5 years ago, I joined Dr. Bellido here at UAMS, this time to study the osteocyte, so the mechanosensor cells and the cells that orchestrate the all bone remodeling process. So as you can see, bone has been my common themes, and I change cells. And where next? So I don't know yet where, but for sure this time my daughter Nina will help me with all the driving. So, well, I'm sure you all know, and just a brief introduction of the disease I will be talking about today, and is diabetes mellitus, and is divided in two main categories. Although there are other clinical manifestation, clinical types of diabetes, and type 1, and the development of type 1 or insulino absolutely deficiency is associated with genetic and environmental factors. And it is usually diagnosed in children and young adults. And although it's not fully understood, it is presumed that there is an activation of immune cells that leads to an immune response with the formation of autoantibody that against the pancreatic beta cells, the cells that produce insulin. And this process therefore induces B-cell destruction and destroy the ability of these cells to synthesize and secrete insulin that leads to the onset of type 1 diabetes. Now, type 2 diabetes develop or non-insulino dependent diabetes, it usually occurs in middle age and older people, and is caused by genetic genetic predisposition, of course, and there is a constitutional factor, including insulin resistance, impaired pancreatic beta cell function, obesity, and oxidative stress. And these factors favor insulin resistance, as the efficiency of skeletal muscle and adipocyte and liver to uptake and utilize glucose decrease. So in order to maintain the normal blood glucose level, now the pancreatic beta cells then try to compensate with an excessive production of insulin. And this is the results in hyperinsulinemia in the beginning of the stage of the diabetes, type 2 diabetes. And this regulation of beta cells and insulin production is also characterized by an impaired glucose tolerance. That means that blood glucose raise beyond normal levels. And now when the disease progresses, there is the insulin that is produced is insufficient, and therefore this leads to the onset of type 2 diabetes. Now, as reported in the Diabetes Atlas in 2021, more than 500 million of adults were worldwide were living with diabetes, and the complication, so the well-known complication of long-term diabetes are of the cardiovascular systems, eyes, kidney, nerves, and of course the diabetic foot syndrome. But recently, also bone has been recognized as a target of hyperglycemia. and especially bone fragility, is an increasingly recognized complication in patients with diabetes. And this is because this patient, diabetic patient, have 32% increase in risk of bone fracture. And this is due to a decrease of bone formation, increased architectural deterioration, and also an impairment in the bone repair process. Now, type 1 diabetes frequently manifests before the peak bone mass is reached, is developed. And this has happened around 30 years of age. And this is due to the absence of the anabolic stimuli, anabolic effects that the insulins induce. And therefore, this is contributed to the development of a lower bone mass. And therefore, type 1 diabetic patients experience more fracture throughout their life compared to non-diabetics. Now, type 2 diabetic patients instead have increased bone fragility despite having normal or even increased BMD. And the recent research has identified a variety of alteration that happen at the level of the matrix, the bone matrix, cellular, at the cellular level, the structural level, that might account, or actually that they account for the reduced bone strength in diabetic patient that is often unrecognized with the DEXA scan. Now here are two examples. Here are a few examples of what happened to the bones in diabetic patient. As you can see here, this arrow indicates a vertebral fracture in a 50-year-old type 2 diabetic patients after a fall from standing high. That means a low impact of fall, not due to a sport injury. And here, as you can see, this arrow indicates the trabecularization of the cortical bone, so the external part of the bone. And in red, there is a measurement of the increased porosity that is manifest in the bones of these type 2 diabetic patients. And also, I would like to show this one because, as you can see here, there is no difference in this type 2 diabetic patient in terms of BMD. However, the structure of the trabecular structure, the callus structure, is highly compromised. So, and why this is important? So, it's important because the incidence of osteoporotic fracture way exceed the annual incidence of heart attack, stroke, and breast cancer. And bone fracture mean high mortality and morbidity with the consequent high health care cost. And osteoporotic fracture in people with diabetes occurs at all sites, but the hip appear to be the particularly susceptible to fracture. And this is the problem. This is what happened after 1 year after a hip fracture. So 80% of the patients that experience a hip fracture are unable to carry out at least one activity of daily living. That means dressing up, having a shower. 60% have difficulty with at least one of these essential ADL, but very importantly striking and scary, I would say, 20% of the patient will die within a year of a hip fracture. So understanding the mechanism that regulate diabetes-induced bone disease is indispensable to prevent and treat bone fragility. Now, in an early study conducted in the laboratory of Dr. Berlido using a type 1 diabetic model induced by streptozotocin that kills the pancreatic beta cells, they have shown that this diabetic animal exhibit decreased bone formation, increased resorption, and increased osteocyte apoptosis, and increased SOS expression. and activation of the PTH receptor signaling by two fragments of the PTH RP, the 137 and the 107, 111, if I remember correctly, restore bone mass and strength in diabetic mice. So, and now this is my project. So a common feature of the bone disease with both type 1 and diabetes is reduced osteoblast number and function and low bone formation. And however, the standard of care are anti-resorptive agents such as biphosphonate or denosumab, which stop the bone loss but do not increase bone formation, and thus failing to repair deteriorated bone architecture. So the current study that we perform, we investigated the effectiveness of 3 bone anabolic agents approved, at least in USA, by the FDA for the treatment of osteoporosis. And is PTH, or teriparatide, that is the parathyroid hormones 134. Then abaloparatide, that is is an analog of the PTH-related peptide 134, and romosozumab, that is the anti-sclerostin antibody approved recently approved by the FDA. And we tested the effectiveness of these 3 treatments in restoring the compromised bone structure using a preclinical model of established type 2 diabetes. So what is PTH? PTH, or parathormone, is secreted by the parathyroid glands in response to a reduced serum calcium concentration. And what it does, it normalizes the calcium levels by enhancing the uptake of calcium from the intestine, by enhancing the reabsorption of calcium from the kidney, and by stimulating, and this is why is important for us, by stimulating the calcium release from the bone. And PTH exerts its anabolic effects by binding on to the PTH1 receptor on target cells of the osteoblastic lineage. And as you can see here, it stimulates the proliferation of mesenchymal stem cells and the commitment of those cells into osteoblastic lineage. And the effect of PTH directly in adult immature osteoblast is to activate anti-apoptotic stimulus and increasing their activity by increasing the mineral apposition and bone formation rate. And in osteocyte, it represses the expression of both sclerostin and, as you can see here, and DKK1. Now, what is sclerostin? Sclerostin is encoded by the SOS gene, expressed in mature matrix-embedded osteocyte. And this mature osteocyte secretes sclerostin that inhibits Wnt pathway, that is one of the master regulators of osteoblastogenesis and bone formation. And the proof that sclerostin is the main regulator of bone formation was shown by study in patients with absence of reduced sclerostin, and this is an example of a patient with Van Buchen disease that presents high bone mass, and in mice by knockout the SOS genes, and this animal presented increased bone mass, high bone mass. And of course, the high bone mass or the increase in bone mass can be also achieved by administering the neutralizing antibody against sclerostin. So now I would like to present you the data we obtained. So first, type 2 diabetes was induced in skeletally mature 3-month-old male mice by a combination of high-fat diet and streptozotocin. So the mice were fed for 4 weeks with high-fat diet, 60% kilocal, 60% of fat, to induce hyperinsulinemia and insulin resistance prior to injection of STZ. And while the control animal were fed throughout the experiment with a normal diet. Now, as you can see here, 4 weeks, now at T2, 4 weeks after the injection of STZ, diabetes was confirmed by blood glucose measurement greater than 250 milligram per deciliter of blood. And also at the same time, at T2, we observe a decrease in total bone mineral density and also, although I'm not showing, decrease in femur bone mineral density and bone mineral density and spinal bone mineral density. It is a measure of bone mass. Now, what we did, we waited an additional 4 weeks, and then at 6 months, we injected the ligands of the PTH1 receptor. And we injected PTH at a dose of 100 nanogram per gram per day. That is the dose that Dr. Belledo used in her previous studies. Dr. Belledo and others used in their previous studies. And then we tested equimolar doses of abaloparatide, the one in pink, or we also tested half of the dose of abaloparatide that correspond to 47.5 nanogram per day, and was already shown to be effective in different animal models. And as you can see here, please stay with me. So the pair indicate in gray is the bar at T3. So in this graph is the total BMD at T3, while in white is the total BMD at the end of the experiment. And as you can see here, at the end of the experiment, the type 2 diabetic mice had a reduced total femoral and spinal BMD that were all corrected by PTH and abalo at low concentration. And importantly, increasing the, what I would like you to notice is that increase the concentration of abalo did not result in an increase in BMD. And this protective effect were not due to a reduction in blood glucose, but it was an effect directly in bone. Now, we also perform the architectural analysis of the cancellus of the distal femur and the cortical mid-diaphysis. So the distal femur is the one close to the knee. And it gives you an idea of the architecture, so the trabecular, how the trabecular are distributed. And as you can see here, we observe a reduction in diabetic animals of BV over TV, so it's the bone volume, the trabecular volume over tissue volume, reduce in bone area over tissue area at the cortical level, so the external part of the bone, and a reduction in the thickness, the cortical thickness, and an increase in cortical porosity. And all these index were corrected in equally restored by the 3 treatments, so by PTH and abalo at both doses, although we observe only a significant increase in BV over TV in the animal treated with abalo. So further analysis it showed that both PTH and abalo increase bone area, cortical bone area, but while PTH was reduced in the marrow area, so there was, let's say, adding bone from the inside, abalo significantly increase periosteal position, so the outside of the bone. And this one, it results in an increase of the polar moment of inertia. That is an index of how the bone is distributed around the central axis, and indicates the resistance of the bone to bending, to breakage. So this micro-CT analysis indicates that abalo may induce more advantageous structural changes. Then we perform 3-point bending. That is an assay on the femur. It's an assay in which the femur undergo a load until the breaking point and measure the strength of the bone. As you can see here, we only detect a reduction in stiffness or rigidity in type 2 diabetes. And 4 weeks of treatment with both PTH and the 2 doses of abalo were equally potent in restoring structural stiffness and also increasing ultimate load or ultimate force and the material ultimate stress. That is the maximum load that the bone can undergo before breaking, and is a measurement of bone strength. However, only abalo, but not PTH, was significantly effective in increase the energy to ultimate load or ultimate force. That is the amount of energy accumulated by the bone before breakage, and toughness. And this is because correlate with the only abalo increase the polar moment of inertia, and indicates that abalo may make the bone more resistant to fracture. We also tested the effect of our treatment of bone serum marker. So as you can see here, diabetes, this is measurement at T3, so before starting the treatment. These animals have significant reduced level of the bone formation marker P1 and P. And both abalo and PTH in green and abalo in blue were, the low doses of abalo were equally potent in increasing P1 and P. Although the high dose of abalo, so equimolar with PTH, elevated P1 and P even further. And this one was consistent with what we found at the tissue level. This is our histomorphometric analysis of the bone formation rate. That is the amount of bone that is deposited in a certain amount of time. And so let's concentrate on the bone formation. So what we see here is the diabetes reduce bone formation, what is called periosteal bone formation, so external bone formation, and endosteal, so towards the bone marrow. And both compounds were not only restore, but further increase bone formation rate. Now, the bone formation results from is the results of 2 measurements. One is the amount of bone covered by the osteoblast, that is measured by the MS over VS, mineralized surface of a bone surface, and the vigor, so how much these osteoblasts that cover the surface, they function, they induce bone formation. That is measured by the mineral position rate. And as you can see here, although both PTH and abalo were significantly increasing the mineral position rate, abalo was inducing it even further. And this one might be the results on increase in P1 and P. Now, this is for the bone formation. Now, regarding bone resorption, what we observe is that before starting the treatment, diabetic animal presented an increased level of the bone resorption marker, serum level of the bone resorption marker CTX. that remain elevated until the end of the experiment and was actually further increased by the treatment with both PTH and abalo. Now, analysis at the tissue level of what we observed was that actually at the end of the experiment, we observed a reduction in osteoclast, and the treatment did not change the number of osteoclast presence on the bones. And this one is the results of an indication of low bone remodeling, that is what it characterizes diabetes in patients. Now, we also perform a bone transcriptome analysis of the L4 vertebra of our animal. And what we found was that a high number of genes were downregulated by type 2 diabetes. Conversely, the majority of genes were upregulated by the treatment, and in particular by abalo, that induced the biggest perturbation on the bone transcriptome. And among the genes that they were downregulated, the gene that is called SCD1, that is the, if I remember correctly, the astearyl coenzyme A desaturase, was previously shown, was the most significantly downregulated. And this gene was previously shown to stimulate osteogenic differentiation and to be downregulated in the bone marrow of type 2 diabetes patients. And abalo was effective in upregulating it. Now, we found that 25 genes were transcriptionally regulated by all treatment. And while PTH exclusively regulate 28, abalo at the same the equimolar doses regulated 326. So again, a higher perturbation of the bone transcriptome. Now we compare the gene expression signature using a different approach, and this is actually what the reviewers recently asked us to do in our paper. And stay with me because I think this is a very important piece of data. So what it does, this analysis is a threshold-free analysis. That means you don't set up any threshold for the analysis of your gene, of the differential regulated genes. And it ranks the genes based on the degree of differentiation, of differential expression in the two groups, in the two treatments. This analysis generates this kind of map that show the strength, the partner, and the degree of correlation between the expression profile of two gene set profile. Now, in this case, PTH and abalo. And what we found is that there was a high concordance in transcriptionally upregulated and downregulated genes between the two treatments, between PTH and abalo. And importantly, none of the genes was, we did not find any discordant transcriptional pattern among our treatment, because these two quadrants were empty. Now, in this map, it's also important to notice the dimension, the size of this quadrant, compare the upregulated compared to the downregulated. And this one shows that the majority of the genes were transcriptional upregulated by the treatment. And then we perform also geo mapping, and we selected gene ontology mapping. We selected three in process: bone formation, bone resorption, and wind signaling. And what we found is that in diabetic bones at the end of the experiment, all three biological processes were downregulated as a confirmation of the low bone remodeling stage of the disease. But importantly, the two treatment, PTH and abalo at both doses, significantly reverse at the same extent the majority of the biological process involving this, the majority of the GO involving this process, this biological process. So this this means that PTH and abalo corrected the diabetic transcriptome signature of low bone remodeling and decreased weight. Now, we also measure SOS expression and sclerostin level, because as I told you before, in our early experiment, we found that bones from diabetic mice had a reduced expression of the osteocyte-derived Wnt inhibitor or Wnt antagonist SOS. Now, here we did not observe in this model, type 2 model, an increase in SOS at the mRNA level, so in the bone, and in sclerostin at the serum level in this animal. However, both PTH and abalo were effective in reducing SOS expression, but only abalo reduced the serum level of sclerostin. So therefore, we next investigate whether sclerostin inhibition per se was sufficient to reverse the bone deterioration induced by type 2 diabetes. And we use the humanized, FDA-approved monoclonal anti-sclerostin antibody romosozumab at the dose of 100 microgram per gram weekly for 4 weeks. And again, we confirm the reproducibility of our model, because as you can see here in this white bar, we observe a reduction in total BMD, a reduction in femoral BMD, and a reduction in spinal BMD. And treatment with sclerostin not only reverse, but further increase all the BMD at the all three sites, both in type 2 diabetes, diabetic mice, and in control animal. Sclerostin treatment also restore effectively, I would say, restore bone microarchitecture, both the cancellous and the cortical bone, and restore porosity. And how it does it is by reducing both the marrow area and increasing the tissue area, and therefore with a consequent increase in polar moment of inertia. The sclerostin antibody also improve both structural and material properties in these diabetic animals, and also in control animal, as you can see here. But what we found, and what is I think this is the piece of data is important, is that as we show in the PTH and abalo study, P1 and P level at the end of the experiment were similar to control level in diabetic mice, and the sclerostin was efficient. The sclerostin antibody was efficient, significantly efficient in increase P1 and P, and as a result, it increased again bone formation rate, and both at the periosteal and endocortical level. But in contrast with the effect of the PTH1 receptor ligands, treatment with the sclerostin antibody markedly reduced the osteoclast number on the surface, and also reduce serum trap, that is a marker of osteoclast number, but we did not detect, although we did not detect a reduction in CTX bone resorption marker in the serum of this animal. So, and this one also happen, so the protection of bone happen despite no correction in glucose level. So this is my last slide, and as you, I hope I convince you that our model is, first of all, is reproducible, and our type 2 diabetic mice exhibit a decrease in BMD and a reduction in bone formation, and an alteration of the bone structure that compromise the bone strength. And all three anabolic agents increase bone formation, but the simultaneous inhibition of bone resorption with the sclerostin antibody leads to a superior bone gain, and so bone mass and strength. And again, just to repeat myself, all anabolic agents protected the bone, protected the skeleton, even in the context of a full-blown hyperglycemia. And with this, I would like to thank you, the source of funding, and all of you for attending my talk. Thank you so much.
Thank you, Silvia. Inspiring, a lot of data here.
Yes.
And yeah, and also very important for patients, I think. So I think we can open the discussion. We have something like 15 minutes of discussion. So you can just raise your hands and ask your question, or you can write it in the chat if you wish. So don't be shy. Try to ask your questions. I can start actually, because these drugs are also used in humans, right? All of them.
All of them
And,
in the United States.
yeah, and in humans you see similar differences according to the mechanism of action. So sclerostin is more potent and doesn't affect. Doesn't increase bone resorption, or in mice is different.
Okay, so although there are no clinical trial directly on type 2 diabetic patients, but the majority of the data derive from post-hoc analysis of previous clinical data. So what they show is that actually abalo compared to PTH induce less bone resorption effect compared to PTH. And this is why in clinics might be more efficient and induce more bone formation. Although this is not with what we observe in the animals, and other publications have shown exactly the same. So PTH abalo in mice work exactly the same. Now, in patients, I think there is a trial that does a direct comparison with sclerostin and PTH, if I remember correctly, and sclerostin was observed to be more, romosozumab was observed to be more efficient. And I think it was published from Beate Lanske and Professor Beate Lanske. And although, again, as I said, in diabetes patient, there is no sort of like a set clinical trial yet.
Yeah, but did you try also to use the anti-resorptive drugs, for instance, with PTH and MBL, or this is not?
Well, we did not. We did not, and I'm actually, I'm very new to diabetes, so I'm my knowledge is not big enough probably, but what the fragility of bone disease the fragility of bone in diabetes happen or may be attributed to AGE. So these advanced glycation end products that from the collagen that get stored and stay in the matrix. Now, and having bone resorption or a physiological bone resorption will eliminate this AG because will contribute to the resorption of the bone, and having a bone anabolic will induce bone gain. So this is why maybe there is a mean of revise the current treatment of bone disease in diabetes patients.
I think that the problem is that PTH affect both bone formation, bone resorption, so it's really a tricky drug. We have a couple of questions here from the audience, from Luca. He says, thank you for your very interesting talk. Maybe I missed it, but how did you perform the administration of SZT and anabolic drugs?
So we injected STZ IP, and the anabolic drugs were injected subcutaneously. So PTH and abalo were injected daily for 28 days, included Saturday and Sunday, so 7 days a week, while the sclerostin antibody was injected once a week, again subcutaneously.
Okay, but the SZT is administered before.
Yes, 1 month, so 4 weeks after IFAD diet. So the animal, this is why it's an established diabetic model. So it's not a preventive. So we actually prove that by using anabolic, we sort of like treat the diabetic disease, because we waited other additional 2 months after injected STZ for the bone disease to occur.
Yeah, which under a translational point of view
Yes.
is more important than
More
the
important,
prevention.
of course.
Yeah. We have a question also from Pier Giorgio. He thank you for the interesting talk. He said, I have just a question. Given the interaction of glucose with osteoblast cells via GLUT receptors, after the anabolic treatment, did you check how the glucose levels in diabetic mice can affect the bone and therefore interfere with the treatment?
So
if there is a crosstalk between.
Yeah, so we perform glucose measurement monthly, and we did not observe any effect of our treatment of glucose. So the effect that we see in bone is independent whether, is independent of the glucose, because the glucose is still very high. So it's a local effect on bone instead of systemic reduction. These drugs are not anti-diabetic drugs. So they solve the bone problem, but they won't solve the diabetics problem. So they would be used in combination with an antidiabetics, because you still have the full-blown hyperglycemia going on, and all the other that we did not study effect on cardiovascular disease and retina and all the other effect, all the other complication in diabetes. So.
Yeah. But still it's important to take care of the bones, because I know people with diabetes and they fracture very easily.
Well, 32% is a huge increase in fracture.
Yeah, and they fracture several times, different bones, so it's really a nightmare for them. So I really understand the point. In terms of treatment with PTH, the analog and so on, what is the vitamin D3 status of this patient, or this mice, sorry. Maybe some comment. And the renal function, generally speaking.
We did not measure vitamin D, so we do not know if they were normal or vitamin D deficient mice. We did not. And we kept the kidney, the kidney, but we did not perform additional testing. So they are in the freezer. I mean, so they were healthy. I mean, no animal was actually this is a 15 animals cohort. And I think, so times all these groups. And I think we had, over a period of 7 months, we have one animal that died before the end of the experiment. So we did not observe any complication, but as I say, we did not perform any histological analysis of the kidney. So I cannot
But
say for sure.
nevertheless, the vitamin D3 pathway and also the relationship with the FGF23 pathway and so on could be something interesting to investigate
Could be,
in this model.
actually is something that is an idea for for the future.
For the future,
yeah. Because also, I mean, as you know, the muscle are very affected. And actually, we observe in the animal, in our diabetic model, we observe loss in muscle mass. We did not perform a contractility analysis, but we measure the bone mass and we observe a reduction in mass. And Dr. Bellido is interested in vitamin D and muscle as well. So another project of the group is, so there might be most likely.
Yeah, every result brings new ideas.
Yes,
correct.
correct.
Never-ending story. Yes, I understand perfectly.
The ideas never are always so many. So the problem is the time and the hands to.
Sure, yes. I have another question regarding when you treated the mice with the anti-sclerostin and you found this effect on bone resorption. Is this a direct effect of sclerostin that was blocked by the antibody, or does it depend on the crosstalk between the osteoblasts and the osteoclasts? Any idea on that?
It does, as you started, it does depend from the crosstalk. So there is no, please correct me if I'm wrong, you as the expert of osteoclast, there is no direct effect of the sclerostin.
Of the sclerostin, yes. In fact, I was curious if there were any new data on that.
No, not that I'm aware, and I think actually, if any, there is the anti-sclerostin will increase Wnt, and Wnt downstream pathway is downstream gene is OPG. So if any, there is an inhibition, but it's not through a direct effect.
Sure. Yes, I understand. A curiosity, you have now a long list of new genes that are regulated. Yes. Is there anyone, I don't want to know which one, but is there anyone very interesting, very strong that you want to investigate further?
Are actually, and this is a project for the next grant, we are actually observe, well, it's well known that diabetes affect or induce a state of an inflammatory state. And so we are actually sort of like, because the effect on bones by those compounds is really well studied. So we are actually sort of like try to open new venue and maybe combining with muscle and bone crosstalk and inflammation. And so this is the idea for the future.
There is a lot, yes.
Yes, there is a lot.
Is there any other question from the audience? Anyone that wants to unmute and ask the question? We have a few minutes more. I don't see any. Are you sure? We have a good opportunity here. Okay, so I think that there are no further questions. Silvia, well, it's a new project for you. It's completely different from the others, but I mean, it seems very, very consistent, very, very strong. And I know that you are in a very strong group as well. So it is easier in this way to have
Correct.
good ideas and support and so on. One question not related to the talk: will you attend the SBMR meeting in Vancouver?
Well, I hope so. If I receive my green card on time,
Okay.
because everything
Because you
takes
cannot go to Canada as well.
exactly to
Wow.
Canada from my immigration status. But I really hope
Okay.
I
But
might.
in case you solve the problem, I hope I can make it. So
Yes,
maybe we can
we can see each
meet
other
there.
after so
Yeah.
many years.
And another thing, just to say hello to everybody there, especially
I will.
Tere. And
Well,
yeah,
she
I
sent her
can.
regards as well. Say,
Yes.
make sure you say to Professor Teti to send to Professor Teti my regards. So for sure I will
No,
make them.
she's really a great scientist, and that has been always a very productive group. So you are lucky to be in that group. Okay, no further question, comments? You want to say goodbye to Silvia? Okay, so thank you, Silvia, very much. It's
Thank you
been
so
very
much.
interesting, and let's keep in touch.
Thank
And
you for the opportunity.
I want just to say to the young people attending here that this is a great example of a person that really is strong, want to do this job, and I think that in the back of her mind, there is also the wish to come back to Italy.
Yes,
And this
the
will
rich.
happen. Italy will be very happy to get you back.
Let's see. Hopefully, when the good opportunity comes.
Yes, but
Yes.
stay tuned because something can
I
happen.
know. I actually be reading that things are happening, things are
Yeah,
changing in Italy. So
sure, exactly,
fingers
exactly.
crossed.
Okay, thanks a lot,
Thank you
and
so much.
yeah, we will keep in touch.
Thank you
Bye
so
bye,
much.
Silvia.
Have a
Bye bye.
wonderful rest of your day. Bye bye.
Bye
Thank
bye.
you.
Bye bye.